Hi everyone, welcome to today's 10 minutes plant talk. I'm Bijun, a second-year PhD student from Dr. Alexander Strong's group in Simsbury Lab. Today, I would like to share my research with you on seeing a plant hormone in action. I would like to start my talk with animal. You're probably familiar with a famous response called fight or flight response. That is a physiological response. When we are in a dangerous situation, our brain tells muscle and nervous system to run away or fight against it. But have you ever feel a rush after this dangerous experience? If you do, this rush is caused by the hormone produced from this fight or flight response. While our brain is making the decision, it is also integrating the signal and trigger a series of hormone signaling. The adrenal gland releases various hormones which act on different parts of our body differently, such as they increase the heart rate, they increase the breathing frequency, and shut down the digestive system. So, what about plants? When plants face a stress environment or stimulation, they cannot run away. Similar to our hormone signaling system, plants produce hormone as well, and they move through the organism. However, unlike in animal, plant does not have gland, and every cell has the ability to produce hormone and transport hormone. Therefore, it becomes important for us to study plant hormone at a cellular level. Plant hormones has different roles on different parts of plants. For example, gibberellin, Ga in short, is important to seed germination, seedling establishment, flower development, vegetative growth of shoot and root. So apart from all these important rules in plant development and growth, why do we want to study Ga? It's also because Ga is important in agriculture. Ga is firstly discovered through fungi which infect rice in Japan. This infected rice grows much taller than the non-infected one, and this disease of rice is referred as silly seedling disease. Apart from rice, Ga also acts on different plants. As you can see in the left picture, the cabbage treated with Ga is much taller than the one which is not. One of the greatest achievements in 20th century is this semi-drop wheat and rice, so we call the Green Revolution. It hugely improved the yield of the crop since more energy is focusing on generating seed rather than elongating the shoot. There are variants of wheat and rice with less Ga produced or lack of response to Ga. Now, we know that there's Ga in plant and they are important. But where is Ga and how much Ga there are in plant? Since Ga has different roles on different parts of plants, we really want to know the detail of where they are and how much they are in the cellular level. So in another way, is how much Ga in this cell? I would like to tell you about the tool developed in our lab, which is used in plants to study Ga in a cellular level, the FRET biosensor. Before I tell you more detail about the FRET biosensor, I would like to introduce you to fluorescence protein. The green fluorescence protein found in jellyfish is said to be fluorescent because this protein glows bright green when exposed to blue or ultraviolet light. If you add GFP all to the plant gene, you will get a green fluorescence plant, a plant which you can see in dark. Isn't it fascinating or isn't it a bit scary? If you look at this amazing picture here, it must be hard to imagine that it's drawn by bacteria-produced fluorescence protein. Apart from GFP, now there are different variations of fluorescence proteins such as YFP and CFP. Rather than the 6 different fluorescence protein you can see from this image, we have more than 600 different variants of fluorescence protein now, and there's more and more coming. So how do we use this fluorescence protein in our fat biosensor? Our sensor have 2 main parts. The parts which bind to GA, and there are 2 different fluorescence proteins linked to this part. When there's no GA binding to the sensor, we can see on the left of this figure, there's more cyan light than less yellow light. If we move to the right of this figure, we can see when GA is binding, we have more yellow light, less cyan light. So the change of color of the sensor indicates whether the GA is binding to the sensor or not. In that case, we can measure the GA level in plants. By putting this amazing tool into plants, we can now study the GA amount in plants. What you are seeing here is a Arabidopsis seedling growing in dark. The left panel is the image from our biosensor, and the right panel is the seedling. Each colorful dots in this image represent a nuclear, which we can see it as representing each cell. And the color of the dots represent the level of GA. The red represent high GA, and the blue represent low GA. We can clearly see at the stem region, there's much higher GA compared to the rest. We note that GA promotes plant growth, so it means that in this region of the seedling, there's a lot growing happening. We can imagine this dark-grown seedling as a young seedling in soil. They want to grow taller to go over the ground and meet the sun. Therefore, GA is accumulating in this stem region to help them grow quickly to go over the ground. The sensor we called NGPS1 is very useful to study cellular distribution of GA. But GA level is very dynamic in plant. It goes up and down very quickly. Therefore, in my project, we are still trying to improve the sensor. For example, one problem this sensor have is they bind to GA very tightly and doesn't let go. Therefore, we try to make change in the sensor by changing the amino acid to loosen the interaction between molecules. So now we have the next generation of NGPS called NGPS2, but we are still on the process of making this sensor better and better, which can give us more accurate measurement of GA in plants. After I have introduced to you about NGPS1, NGPS2, and other potential NGPS, we can now investigate the GA distribution in cellular level in plant. However, there are different compartments in cells which have various conditions. For example, in some compartment, the pH is very low, which means that it's very acidic. Our sensor is no longer functional in this acidic environment. Therefore, we are trying to resolve it by making a new sensor with different pH resistance fluorescence protein. In conclusion, our research aims to address the question of where is GA, when is the GA be there, how much GA there are. And on the side, we are also trying to improve our biosensor to allow more accurate investigation of this question. Also, in all these different plants, including the important crops for our society, how is GA level regulate the shape of them is an important question to answer. Hopefully, on one day, it can help us. Answering this question can help us to design our crop with more desirable characters. At the end of my talk, I would like to thank my supervisor, Dr. Alexander Jones, my mentor, Dr. Annalisa Rizas, and everyone from the Jones Group. Thank you very much for listening to my talk and hope you have a nice day.